Yeah, so up until recently, our work was really guided by what's known as sort of the thermodynamic hypothesis of protein folding, which is that proteins fold to their lowest free energy states. And so in that picture, if you want to predict the structure of a protein, you start with the amino acid sequence, and it basically is a search problem. It's a global optimization problem. You're trying to find the lowest free energy state. And if you want to design a protein which folds up to a new structure, you start with that structure and you search for a sequence whose lowest free energy state is that structure. And I'm assuming everyone's familiar with the basic idea that amino acid sequences, that structure of proteins is encoded by their amino acid sequences. And so the basic idea is that the way that coding works is that the lowest free energy state for a protein sequence is its folded structure. So in this picture, if you want to do either of these problems, whether it is predict the structure of an already existing sequence or design a new sequence that will fold up to a new structure, the problems basically the problems are first of all one of sampling through the very large number of possible structures and sequences, and then evaluating energies. And we have developed a program called Rosetta over the years for calculating energies to do these problems. So just to give you a feeling for the size of the spaces that are involved, there are, even if you just restrict to the 20 naturally occurring amino acids, their 100 residue protein will have on the order of 10 to 130th possible sequences. And this really dwarfs the number of sequences which have existed in nature. And so if this diagram depicts all of sequence space, native protein sequences, the ones that exist in nature, are really a tiny, tiny fraction of the possibilities. And they're represented by these red islands. Most protein engineering, when people have tried to create new proteins for various biotechnological applications, they've taken naturally occurring proteins and made very small numbers of changes to them. So what we're going to be talking about today is making up completely brand new proteins that aren't related to naturally occurring ones. And I'll illustrate this first in the context of what we've been doing to combat COVID-19. And so probably everyone's familiar with the fact that on the surface of the virus, there's a protein called the spike protein, which has a receptor binding domain shown here. And it binds to a protein on the surface of our cells called ACE2. And so this is a picture of the structure of ACE2 bound to the receptor binding domain. And so the basic goal here was to design a protein which would bind more tightly to the surface than ACE2 does. So it would basically block that site and prevent and not allow the virus to infect cells. So we did it in two different ways. One was to take a piece of ACE2 and to build a brand new protein around it, as shown here. The second way is to make up very large numbers of proteins on the computer and then basically dock these against the surface computationally, design the interface so it's shape complementary to the receptor binding domain. And then in either case, what we get out are just linear amino acid sequences. And they encode these hypothetical proteins which are predicted to bind tightly to the virus protein. So the next step is to encode these. Actually, I'll show you some pictures here. So this is sort of what these things look like. They're the ribbon. The ribbon pictures up here are the design proteins, and they're quite small as proteins go. This one just has 55 amino acids. It's very stable. You can boil it, and it's fine. So at this point, this is kind of science fiction because we just have the sequences, but we haven't made them yet. To actually make the proteins, we take advantage of the fact that it's very the world's worked out a very efficient and cheap ways to manufacture DNA. So the genetic code takes you from DNA sequence to protein sequence. So we can basically make use of the genetic code to encode, to synthesize pieces of DNA that encode these brand new proteins. And we can actually do this in very large scale. We can encode up to 100,000 different designs in DNA and then test them all for binding against the virus. And we found a number of proteins which bind very tightly to the virus. So here is the electron microscopy structure of one of these. So this triangle object here shown, this is a top view and this is a side view. This is the viral spike protein. And this little ribbon thing is our design protein. So first of all, you can see that it's binding to the virus, and there are the spike protein is a trimer, so there's 3 copies that bind. And if you now compare this experimentally determined structure with our computational model, they're superimposed here in these 2 different colors. You can see they're basically identical. And if you look at even at the placement of the side chains, they're very, very similar. So this shows that we can basically take an arbitrary sort of target surface and now design proteins which bind to it tightly. So we've done quite a few experiments showing that our collaborators have, showing that these proteins protect animals from infection from the virus. And because it's so stable, you can just spray it into the nose, and there's very substantial protection from quite a bit before to after infection with the virus. So I showed you that there were 3 copies of this bound, and we can take advantage of the symmetry. Sorry, this is a little bit hard to see, but here's the spike protein again. Actually, it's maybe clearest over here. We can design symmetry matched arrangements where we have these 3 binding domains that are lined up with 3 receptor binding domains. In this case, we get sort of the free energies add, because you're getting all 3 sites engaged at the same time. So this becomes nearly irreversible binding, and it's very effective. These compounds are very effective against all of the variants of the virus we've tested, including the Delta variant, which is spreading like crazy now here. This is just showing that these, again, protect animals against all the different variants. So another neat thing about these proteins is that you can produce them in very high levels, and they're very stable. And so this is where we were a number of months ago. And so we got very excited about these as potential therapeutics, because you could make them in very high levels. They're much cheaper and easier to produce than antibodies. So they're slowly going through the clinical development process now. But we can take these methods and now make binders to proteins that bind to proteins on the surface of our own cells, like growth factor receptors. And we can do this quite robustly. And so we're quite excited about this, providing sort of a completely new way to control signaling in biological systems, because we can bring together receptor subunits in new ways using the designed proteins. So the second problem that I want to talk about is the problem of detecting the virus. So what I described so far was the design of these small proteins which bind very tightly, but we can also design proteins which have multiple states. So this is like a molecular device. So the system has 2 components shown here. This is in the closed state in the absence of the viral protein. In the presence of the virus, the thermodynamics of binding of this element within this system drives opening of the cage. And this binding domain is the same one I just described you before. So here what we've done is couple the thermodynamics of binding of this small protein to the target to the opening of this switchable system. And the way we've configured it in the closed state, in the absence of the virus, it's dark, but when it opens up, it emits light. And there's sort of an illustration of how that works here. And so this is just looking at luminescence as a function of time. And in the absence of the viral protein, it's dark. And when the viral protein is added, there's light is emitted. We can use this now to monitor the responses that people make after vaccination, because this system is in thermodynamic equilibrium. So it's in the dark state. We add the viral receptor binding domain and it opens. And then if we add a serum sample that contains an antibody against the RBD, it competes it away from the sensor. and we get a decrease in signal. And so we found quite good correlations between the extent of neutralizing antibodies in a person's serum, which take quite a while to measure, and the change in the sensor signal when you add that sample to the sensor. And this is nice because it's almost instantaneous, the measurement, and you just need to monitor the light emitted. And we've been able to use this system to make molecular devices for sensing a wide variety of compounds. I think I'll just skip through this. But we can use the same concept of designable switches to do other sorts of things. And for example, this illustrates the performing of logic, doing logic on the protein cell surface, on the cell surface. So in a number of situations, like in cancer, you want to be able to detect cells that have combinations of surface markers rather than just one single marker. For example, most tumors arise from normal cells. They don't really differ that much from normal cells. So you might want to, so it might be that there's no single marker that distinguishes tumor cells from normal cells, but say there's two markers that together distinguish. So the way we can do this is similar to what I just described. We can have one component of the system be directed to bind to one marker, the other component bind to another marker. And only when they're both there does the system open, recruiting an exposing element that recruits the system, which then kills the cell. And probably some of you have heard of CAR T cells, and these are cells which are programmed to kill cells that are recognized by these T cells that you put into the body. So we can make T cells that recognize the epitope that's exposed here. And what we find, and this is a little bit more sophisticated logic, this is A and B, not C, because we put also on the cells something that sort of shuts down the system, and that's sort of like a healthy cell marker. In this case, we only get killing of the cells that have markers A and B, but not A, B, and C, or just A or B. The final area I'll tell you about is vaccines. And so we've been working for a number of years on methods for designing self-assembling systems, protein systems. So the basic idea here, this is illustrating the design of icosahedral structures. We start with protein objects of 5-fold and 3-fold symmetry, and we put them on the 5-fold and 3-fold symmetry axis of an icosahedron. Then there are basically 2 degrees of freedom for each one. They can spin around their symmetry axis or move in and out, and that preserves icosahedral symmetry. And then basically we design interfaces between them, and then we make the proteins in the lab. And we can do this now very robustly. So the proteins come together and form these really beautiful icosahedral particles. So my colleague Neil King at the Institute for Protein Design took the receptor binding domain of the spike protein and put it on the surface of one of these designed icosahedral nanoparticles. And that's shown here, these little bumpy particles. These turn out to be extremely potent. I'm not going to go through this slide in detail, but these elicit extremely strong responses against the virus. So if animals which get these make very strong responses, and they're currently they're in pretty advanced clinical trials now. The results look really good, and so it's very likely that these will actually be out in the world for use. They give much stronger responses than current vaccines do, and they can also be manufactured on a much wider level. They don't require the very cold temperatures that the mRNA vaccines, for example, take. So we can basically take this concept of matching the point group symmetries of regular polyhedra to make other types of nanoparticles. So for example, this is one where we take antibodies, which are widely used in nanotechnology, and put them on the C2 axis, for example, in this case of an icosahedron in the middle of an edge. And we design another protein that sits on the 5-fold symmetry axis. And these together will assemble into form very uniform nanoparticles, but now these have antibodies sticking out, which turns out to be very interesting for a wide variety of biotechnology applications. And we can also, sort of thinking beyond medical applications, we can design proteins which bind chlorophyll dimers, the same chlorophyll dimer in the same arrangement as in sort of the photosystem, that's the heart of photosynthesis. And then we can take those dimers and build them, in this case, into octahedral, sorry. No, I can only go into octahedral structures. And you see these very beautiful octahedra here, which have 2 chlorophyll molecules in the middle of each edge. And so now what we're trying to do is to put so we've got sort of like a synthetic light harvesting complex. And now the question is, what can we do with all the light that's captured? We can also design proteins which aren't free in solution, but actually go to the membrane and insert in and form channels. So, for example, here's a design protein which selectively allows potassium ions to traverse a bilayer. So here on the outside of the protein, we've put nonpolar residues that are comfortable in membranes, and then on the inside we have a water-soluble a channel lined with polar groups, and so we can make these synthetic channels now. We can also make proteins with larger pores that are in membranes, and so here this is a protein made out of beta strands, which has a sizable pore in the middle. This is the design model, and this is the crystal structure. And we can now make these pores bigger and bigger, and this is very interesting for a pretty wide range of applications, ranging from DNA sequencing to protein sequencing to just simply selective filtration, because we modify the lining of these pores. And then sort of the final example I'll give you is the design of protein rotary machines. So here we are designing 2 classes of proteins, a axle, which is sort of a linear protein shown here, and a ring, which fits around the axle. And we've worked out ways for assembling these types of structures, these rings around the axles. And it's really kind of neat. You can look at the energy landscapes as you rotate the ring around the axle, and there are different cases. They're the case where the axle and the ring have the same symmetry, so C3 symmetry. So here there's 3 clear rotational minima. But in other cases, we can when the symmetry doesn't match. So here we have a dihedral D3 axle and a C5 rotor, and you can see that the energy landscape is much rougher. And when we look at these systems by electron microscopy, we see much more blurred out density, suggesting sort of different rotational states in this case than in the C3 case. Now, I want to show you a little bit more on this one, which is a D8 axle on which we've mounted 2 different rotors. And in this case, we can actually distinguish 2 different rotational states. You can see that the 2 rings are offset relative to each other, looking down the axle here, and aligned in this case. And so this is really exciting. Now, obviously, the challenges are to be able to visualize the rotation, and to be able to ultimately drive it with chemical or light energy, which is what we're working on now. So everything I described so far, it was sort of with this physically based model, but as you're well aware, deep learning is becoming more and more powerful. So inspired by real progress at DeepMind, we've been developing networks that basically take sequence and predict structure. In this particular network, we're processing informations at the sort of 1-dimensional sequence information, the 2-dimensional sort of residue interaction map level, and the 3-dimensional structure level. And information is flowing back and forth between these at all levels. And this method is quite good, much better than what we had before. It's not as accurate yet as AlphaFold, the DeepMind method, which is really impressive. But we've been able to use it to solve quite a number of hard problems, and we can also use it to predict the structures of protein-protein complexes, which carry out a lot of biological function. But what I want to spend a little bit more time here is the use of this sort of network to design brand new proteins. And this is really a quite different picture than sort of the energy-based methods I've described so far. So I basically, what these networks do is they take an amino acid sequence and produce a three-dimensional structure. And basically what you can do is instead feed in a random amino acid sequence and simply assess the extent to which a single structure is predicted, and then do Monte Carlo optimization or gradient-based optimization to maximize the extent to which the sequence is predicted to fold to a structure, basically any structure. And when you do this, this is looking at basically an interaction map between pairs of residues. So you start with a random sequence. There are very few predicted contacts here. That's the off-diagonal regions here. But as you go through a Monte Carlo simulation, contacts grow in because basically you're rewarding that. And in the end, you end up with sequences that are predicted to fold into very precise structures. And we can make these proteins in the lab. These are now, we call them hallucinated proteins because they're like kind of like the images of ideal cats you get if you take a network that has been trained to recognize cats, and then you ask it to, that you start with random images and you optimize for the network to predict them to be cats. In the same way that these proteins just are sort of the network's idea of what an ideal protein looks like. And we've actually solved the structures of a number of these proteins, and they come out quite similarly to the hallucinated structures. So we can now take this method and put in constraints to design proteins that contain particular types of active sites. So we can not only reward sort of overall folding to some structure, but the presence of some particular structural motif. And so, for example, this is a large protein involved in the immune cascade, and it interacts with its partner just through a small region here. We can now carry out the hallucination process and just reward formation of the of this small part, and we get different solutions as shown here. We can take insulin, for example, which has a very complex fold, and mimic this in small proteins. So okay, well, I think I went through kind of quickly, but I think we have lots of time for questions. So the work I described, let me give the acknowledgements, the most important part. Longjing Cao and Brian Coventry designed the COVID antivirals. We had a lot of collaborators on all the animal experiments. The sensor work was done by Alfredo Rubio and NDA. The vaccine work was from Neil King's group, as I described. The de novo membrane proteins was work by Chunfu Zhu and Peilong Liu. The rotary motor work is done by Alexis Corbet. And the structure prediction work is by Linkyung Bak and Frank DeMaio. And this folding is designed by network hallucination is work by Yvonne and Sam and Tamuka, and Doug and Ju and Sydney are doing the work with the constrained hallucination. So why don't I stop there and be happy to take questions on any of this.
Okay, thank you for the interesting talk. Quite impressive what is possible. I have also some questions, but first other people. So first one was Martino.
Hello. Hi, Martino Bolognesi from Milano, Italy. Okay. I actually have two quick questions. The first one concerns the design of RBD binding proteins. You hear me?
Yes.
Okay, good. Essentially, what you are proposing is a sort of shield against the virus. So it's not really a drug that will kill the virus. Do you envisage any way to turn it into a virus-killing drug? The second question. Second question, I'll be quick. In these hallucination proteins that you design, do you find by chance any new protein topology?
Yeah, those are both really good questions. As far as the first one goes, these proteins will bind very tightly to the virus, so you could attach to them some sort of chemical or other protein like a protease that would destroy the virus. We haven't done that, but I think it's a good idea. Second, it's a good question. It sort of depends on how you define topology. In general, what the proteins that come out look sort of like idealized versions of naturally occurring proteins. They don't look as different from naturally occurring proteins as the things that we make by sort of intentional design.
Okay, thanks.
Next question, Alvaro.
Hi, thank you for a fascinating talk. I have a very technical question, which is, how do you make your gorgeous diagrams?
Well, I had nothing to do with that. There's been a lot of work on software for protein visualization. I think the program we use is an openly available program called PyMol. But yeah, that's the real miracle, isn't it?
Yes, thanks so much.
Okay, typical Alvaro question. Gabriele?
Yes, thank you so much for the amazing talk. I have a pretty naive question because I admit I really don't know a lot of the things we were talking about, but it's about let's say these molecules or these proteins that you would use, for example, for cancer, but also the ones that you were talking. In the case of the tests that you did, for example, with animals, it sounded like you would just spray some of these through their nose. But in the case of cancer, how would you deliver these through the body?
Yeah. So for cancer, you would do it one of two ways. You would either it would be like a drug that's administered by injection. So it would be injected into circulation, or in some cases injected into the tumor itself.
And so is there any constraint that comes from the fact that it going through the body, your body might do something with it or something? So is there a constraint that you need to be aware?
Well, yeah, so there's always that's why drugs are so expensive, because of the clinical trials and safety tests that are required and I think these de novo designed proteins are like any other drug. You have to do all the safety tests. So far, in the limited, we have a company that's developing one of these designed proteins as a cancer therapeutic. So far, there haven't been any safety issues that have, but the body is very complicated, so there's always a possibility that there's some interaction that you didn't think that was not expected. We haven't seen that so far, but it's always possible.
Thank you.
Next is Marek.
See, I can't really hear.
Can you hear me now?
A little bit, yes. Talk loudly.
Okay, I wanted to ask about...
You must speak louder.
I wanted to ask about diseases which are caused by prions. Can you hear me?
Yeah.
So, can one do something about these diseases with this approach?
Yeah, so we're trying to design proteins now which bind to the monomeric. These are proteins, these are diseases caused by association of proteins into big amyloid fibers or other types of fibers. And so one can try and design proteins which bind to the monomers and prevent them from associating, like the A beta and tau proteins that are involved in Alzheimer's, for example. So that's an approach we're taking. Whether it will be useful or not is just too soon to say.
So there have been no animal studies yet?
No.
Okay, thank you. Very interesting.
Okay, thank you. Next one is, Heori.
Thank you very much for the lecture. I'm not a particle physicist. I'm from University of Geneva Department of Organic Chemistry. I was wondering, you mentioned self-assembly of proteins. It is a supramolecular structure, and how did you make it? Was it a template or something else?
Yeah, so in this case, it's So the icosahedral particles I described are made out of 60 copies of 2 different proteins, and we simply encode those proteins in synthetic genes, and we make the proteins in bacteria, and they spontaneously assemble. This again, it's the same principle. We're designing the lowest free energy state of the system are these 2-component icosahedral particles. So it's all spontaneous self-assembly.
And I was wondering, what is this potassium's like channel which spans the membrane? It's like it's special for potassium because it has like polynomycin-like structure, or?
It's a good question. We actually did not design it to be. We just were trying to design transmembrane pores. It's probably because potassium has the hydration layer is not as tightly bound in potassium, so it can go through the channel more readily than sodium, for example. But now we're trying to change the lining of the channel to see how that affects conductance. So this was sort of a property, an emergent property we didn't design in.
Okay. Did you study maybe it's like a potentiometric or electroneutral transport occurring?
No, I mean, I think there's a lot of things to do, but we basically just actually our collaborators, we design the proteins. Often doing the more detailed measurements is beyond our abilities.
Okay, thank you very much.
Are there other questions or comments? Okay, so I have 2 questions about finding a sort of the minimum energy. First of all, there's a computational problem, of course, but isn't it also the time evolution would be important? For example, these foldings are non-commutative, so probably one folding has to come first, and then the other one happens, and then only then the energy is lowered. So it could be sort of that the order of the foldings plays a role, and how do you simulate this with a computer?
Yeah, well, it's a very interesting point.
Of each step, yeah.
Yeah. So basically, before our work, you could have, it seemed plausible that the amino acid sequences of proteins were optimized not only to have the correct folded structure be the lowest free energy, but also the sort of the sequence of events somehow programmed in. But in our design approach, we're purely considering the thermodynamics. There's nothing about process. So we're not doing any design for dynamics or for order of events. And our proteins fold, our designs fold very rapidly. So it suggests that, the standard picture that people have as you make interactions, that the free energy drops. And so you get sort of this funnel-like landscape. So I think basically that the fact that you can design proteins only focusing on making the design target state very low in free energy. So the fact that that's sufficient to drive folding is sort of an argument that that the events, these molecular conformational changes, are fast enough that that despite the intuition that it should be very slow in practice, it's if as long as there's a deep enough minimum, the system will find it.
So, and the second question, you're probably not just looking for the minimum energy configuration with your computer program. I assume that you use machine learning, and then there are sort of implicit rules like if this happens and this, then there's a tendency for this amino acid to produce this kind of structure and so on. So is it really so that you really go in the landscape and look for minima?
Yeah. So the in the second approach I described, it's pure pattern recognition, right? This design using deep learning. There's no physical model. But in the first case, it's basically I didn't really describe how we do it. You make up a structure on the computer, and then you search through the space of possible sequences. It's basically discrete optimization, finding the sequence which has lowest energy in that structure. Then we basically take that sequence and we do a folding simulation and confirm that the lowest energy state is the design state. So that is actually how we're doing it.
Okay. So last question. Would a quantum computer help here? I mean, the problem is typical for quantum computers.
Yeah, it could. The problem is there's both discrete and continuous degrees of freedom. So the sequence itself is discrete, and the side chains to some extent have discrete conformations. but the protein chain itself is continuous. And so, you know, I'm sure, you know, as quantum computers become more, they will become powerful here. But so far, the sorts of problems which have been looked at in the protein space are more discrete optimization problems, where, you know, you can enumerate some set of states and do calculations on them. So I think there are problems here where quantum computers will be relevant, but I think there are challenges as well.
Right. Yeah. Of course, the whole thing is a quantum computer. Nothing else.
Yes, that's right. It's doing. I mean, that's the thing, right? It is a quantum computer, right?
Yeah. So
it's just some large molecule.
As a simple isomorphic system you can use instead. Yeah.
Yeah.
Okay.
Absolutely.
So, are there any other questions?
I had one quickly, if I could ask, and it's about the actual, you know, shifting to, say, manufacturing for, you know, the technology for doing this at a scale, or any of these things. Is it you know, is it already there? Let's say if you were to have something like you were talking about, that there are some trials and things. So if you were to say, oh, we want to produce this.
Well, yeah. So I mean, so basically, the industry standard drugs are antibodies, and there's very good systems in place for making those in large scale. I told you that our compounds, since you can make them in bacteria, are much cheaper to produce, and there are companies which have set up very large facilities for making proteins that are made in bacteria. They aren't quite as widespread. So there are, we don't have to build anything up. There are resources, you know, in the world for doing this. But it's kind of a novel therapeutic modality, the idea of a designed protein you spray into your nose. So there are more unknowns, not so much on the manufacturing side, but then on the sort of the clinical testing side.
Okay, thank you. Stefan?
Me?
Yes.
Stephen? Hi. I have two rather broad questions. One is, and I'm surprised that physicists haven't brought this up, are there ethical implications? Are there ethical rules or oversight that apply to this? And it's a fantastically powerful technology. And then let me ask you the other side of the same question. What are you doing about climate change? In the sense that could some of your techniques be applied to issues there? Thank you.
Yeah. Well, let's see. So the first is, you know, there hasn't been a lot yet of, as far as the sort of like regulation. It's a very, very new technology. Most of the stuff I showed you was done in the last several years. And also, you know, nature already has come up with some really, you know, really awful things, like, you know, things like the coronavirus or Ebola virus, which I think it's the dangers that we face from designed proteins are far less than the dangers we face from what nature's already come up with. Doesn't mean that it's not an important thing to think about. Climate change, well, let's see. So the problems are carbon fixation, which, as you know, nature does not very efficiently. And then energy capture. I mentioned photosynthesis briefly. One of the real problems is scale, because to have an impact on climate change, you really need something that's going to be deployed at a very, very large scale. And so that means you will have to be likely, you can't make enough, you make enough protein to be a drug or a vaccine, but you can't make enough protein out that will do stuff sort of freestanding. So it's probably going to have to be in a genetically modified organism. And so we're trying to make, for example, proteins which expand the range of wavelengths that chlorophyll absorbs. We've played with alternative carbon fixation pathways, but there's, you know, we don't have anything that's better at this stage. So I think it's obviously a critically important problem, and we would very much like to contribute to it.
Thank you.
All right. Are there other questions or comments? Yeah. FT here.
Hello. Thank you for the talk. I wanted to ask you if this method could be used in in order to trace maybe the origins of life on Earth, or if you, by using it, you could find some primordial proteins maybe, because it's low energy. So
yeah, it's an interesting question. So we can speculate about the origins of life on Earth based on these approaches, like looking at what sort of proteins are most likely to sort of come about by random chance. But of course, that's different from, so we can sort of make general statements, but in terms of actually figuring out what happened, that requires, biological archaeology.
another question is whether you could use that technology to produce maybe microorganisms that can survive in multi-extreme conditions, especially outside Earth, because currently space travel and all these space.
Well, the problem is that you'd have to improve all the proteins in the microbe, right? Because, there's a lot of different proteins. So we can make individual proteins more stabilized, but re-engineering the whole, that would require a lot of expertise in genome engineering, but I guess in principle it's possible.
Okay, thank you.
Xavier.
Hi, David, that was a very interesting talk, thank you. So I have a question from one of your first slides, which is, so essentially what you can do is you can explore areas in the protein space that are not explored by nature, essentially, right? Or by what we find ourselves in nature. So do we understand why certain areas have been more explored by nature, and why these empty spaces exist? Well,
I think it's really just a numbers argument. So the space is astronomical with 10 to the 130th sequences. And nature, evolution proceeds incrementally, right? Because you have some protein or gene and it mutates and so evolution is concerned with function and fitness, but not with completeness. So, it's exploring this space, and it's probably very historical what actually gets found. It only has to be good enough. It doesn't really care about exploration for the sake of exploration. So I think it's really just that the space is so big, and there was nothing that kept nature from finding these other things. It's just that it didn't need them or didn't have enough time.
Thanks.
Interesting. Further questions or comments? Well, if not, then let's think. No, there's another one.
Okay.
I cannot read the name fully, but it's called, well, Evangelia.
Evangelia, yes, that's right. Thank you for the great talk. I'm a structural biologist from the National Hellenic Research Foundation. I would like to ask you whether you have considered using the different rotor structures that you presented as a scaffold for enzymes of biotechnological interest, because this could be like, a bioreactor, let's say, of having different enzymes docked there and be able to act as biocatalysts then.
Yeah, I think that would be really neat. We're hoping people will be interested in using them for things like that. But yeah, and it may be happening. I mean, we never know what happens when we publish papers. But if you know anyone who'd be interested, definitely let me know.
Okay, thank you.
All right. Well, very nice to talk to everybody. I think I have a few minutes to get breakfast, so I think I'm getting a little hungry. So, but those were great questions. I was worried that my talk wasn't really at the right level, but I guess from the questions, it seems to, those were all really excellent.
No, I thought it was just right because, people don't know the background, but just the pictures were clear enough, and then the idea came over, I think. So
Great.
Thanks a lot.
Okay. All right. Yeah. Goodbye.
And have a good breakfast.
Have a nice breakfast.
Okay.
Yeah.
Bye.
Thank you. Bye-bye.
Thank you. Bye-bye.
Thank you. Bye.